If you were to just look out at a peaceful pasture, like just a flock of sheep grazing, you generally expect a certain kind of behavior, right?
Yeah, exactly. They're docile, they're predictable, just, munching on grass.
Right. But imagine if, right in the middle of that super serene picture, one of those sheep suddenly grew fangs, turned around, and just started hunting the rest of the flock.
I mean, that sounds like a literal horror movie.
It totally does. But that exact kind of evolutionary leap from a peaceful grazer to a territorial killer is actually happening right under our noses, just on a microscopic scale.
Yeah, it completely challenges our assumptions about how hardwired behavior actually is.
Welcome back to our ongoing deep dive series into the fascinating microscopic world of worms. Since you, our awesome listener, have been following along, you are already intimately familiar with the humble, docile C. elegans.
Right, our standard laboratory worm. The peaceful sheep, if you will.
Exactly. But today we are looking at a stack of brand new research detailing its aggressive predatory cousin, Pristionchus pacificus.
It's a very different animal.
Oh, yeah. So our mission today is to uncover exactly how a brain evolves a completely new behavior, like specifically how an organism turns into a killer. We'll be unpacking a Nature paper that tracks the exact molecular switches that make P. pacificus so deadly.
Yeah, and before we really get into it, if we could just ask a small favor.
Oh, right. Since you are already part of this awesome worm-loving community, please take a quick second to follow, like, comment, and share this deep dive. It really helps us grow.
It definitely does. So the primary source we are looking at today is this incredibly comprehensive study. They map the evolution of invertebrate aggression by tracking nematode behavior. And they used machine learning and genetic manipulation to do it.
Okay, let's unpack this because tracking a microscopic hunt sounds basically impossible. We're talking about an animal that has evolved literal teeth-like armaments, right?
Yeah, tiny teeth in its mouth.
Just to puncture prey, like the larvae of our peaceful friends C. elegans and feed on their innards. But how do you even track a microscopic hunt, like objectively measure aggression?
Well, that was the first major hurdle for the researchers. I mean, you can't just strap a tiny GoPro to a worm's head.
Right, no tiny cameras.
So nematodes feed using this neuromuscular organ called the pharynx. It's basically a biological pump right in their head.
Okay, pump.
Yeah, and to track when the worm was attacking, they engineered a specific genetic tag. It's called pipa-2p.rfp.
Rolls right off the tongue.
Yeah, well, what it does is it makes the feeding muscles of the predator actually glow red under a special microscope.
Oh, wow. So they basically created a glowing red beacon inside the worm's jaw.
They kind of had to, honestly. You simply can't see those subtle muscle twitches otherwise.
Yeah.
But by tracking that glowing red pump, they could continuously monitor its speed, its reversals, and every single feeding event.
But wait, that must generate like a massive amount of raw data.
Oh, an unbelievable amount. Millions of tiny movements.
And a human just looking at a massive spreadsheet of muscle twitches wouldn't know if the worm was actively hunting or just, wandering around aimlessly.
Exactly. Which is why they handed all that raw data over to a machine learning model.
Nice.
Yeah, humans have bias, right? We see two worms bump into each other and we think, oh, they're fighting.
Right, when they might just be turning around in an awkward hallway, basically.
Exactly. But the AI doesn't have that bias. It used dimensionality reduction and clustering to look at the velocity, the pumping rate, and the angles of the head swings.
So it basically identified patterns we couldn't see.
Yep. And it identified 6 distinct behavioral states.
6 states. Okay, what were they?
Well, some were standard, like the roaming and dwelling states that you see all the time in the peaceful C. elegans.
Like when it's just wandering around eating standard bacteria.
Right. But 3 of the states were brand new and completely predatory. The AI categorized them as predatory search, predatory biting, and predatory feeding.
Predatory search, that sounds super specific, but here's where I get a little stuck, right? Because I think about my smartwatch tracking my movements.
Okay. Yeah.
My watch knows if my heart rate is up and my arms are moving, but it doesn't know why. So are these states just a simple hungry versus full switch? Like my smartwatch data knows I'm power walking to the fridge for a snack.
Right. Foraging, basically.
Yeah. Or is there actual spite involved here? Like the watch shows I'm actively getting into a boxing match with my roommate.
That's a great analogy. And the researchers actually untangled that exact question. They needed to prove that the biting wasn't just indiscriminate eating, but like actual targeted aggression.
So how did they prove that?
Well, they look at how environmental context modulates the drive. When they placed P. pacificus on just a plain lawn of OP50 bacteria, the AI showed them just roaming and dwelling.
It's normal grazing behavior.
Yeah. But the moment they were placed on a plate that had C. elegans larvae, they switched immediately into the predatory states.
Complete with those exaggerated head swings they mentioned in the study.
Exactly. And what's fascinating here is that the researchers noticed the worms bite for two completely different reasons.
Okay, two reasons.
Well, sometimes they bite and then follow it up with predatory feeding. They puncture the prey and eat the innards.
Okay, gross. But that makes sense nutritionally. Just a hungry worm.
Right. But other times, they exhibit predatory biting without the feeding state.
Wait, really? They just bite the other worm and, leave it there?
Yeah, they just attack without consuming the calories.
That feels incredibly aggressive. Why expend the energy to attack if you aren't going to get a meal out of it?
To prove it was territorial, the researchers set up this brilliant experiment. They offered the predators an abundance of bacteria and the C. elegans larvae at the exact same time.
Oh, like an all-you-can-eat salad bar right next to the prey.
Exactly. Zero effort required for the calories. And the worms spent significantly less time in the predatory feeding state.
Because they were getting full on the easy bacteria.
Right, but they kept biting the larvae. The predatory biting state remained super high. They were actively establishing territory and removing competitors from their food.
Wow. For you listening, just think about what a massive evolutionary leap that is. We are moving from basic foraging, just eating whatever is in front of you, to complex territorial warfare.
It's no longer just about calories. It's about control of the environment.
They aren't just hungry, they're hangry, and they're displacing rivals. But that brings us to the neurochemical tug-of-war, the gas and the brake, essentially.
Yeah, what actually flips that switch in their tiny brains?
Right. So in our peaceful C. elegans, dopamine and serotonin generally control foraging. Then you have octopamine, which signals a fasting state, and tyramine, which triggers a flight or escape response.
If we connect this to the bigger picture, evolution rarely invents completely new chemicals from scratch. It's way more efficient to just repurpose what's already there.
And P. pacificus totally rewired the function of octopamine and tyramine, right?
It did. The researchers used CRISPR-Cas9 to actually mutate the worms.
Using those molecular scissors to snip out genes.
Exactly. First, they knocked out the TBH1 gene. That's the one responsible for producing octopamine.
And what happened?
The worms completely lost their aggressive edge. Predatory biting dropped massively.
So octopamine is the gas pedal for aggression. You need it to want to attack.
Right. But here's the wild part. Tyramine and octopamine are synthesized in the same pathway. You need an enzyme, TDC1, to make both of them.
Okay, so if you knock out TDC1, you get zero octopamine and zero tyramine, right?
Yes. And you would logically assume that a worm with no gas pedal would just be super peaceful.
Totally docile, yeah.
But the behavior showed the exact opposite. When they knocked out TDC1, meaning both chemicals were gone, the worms were highly aggressive again.
Wait, let me make sure I'm following this. If you take away the gas pedal, the car stops. But if you take away the gas pedal and this other thing, the car suddenly speeds up again.
Yeah, which tells us exactly what that other thing, tyramine, is doing. Tyramine acts as the brake pedal.
Oh.
Without tyramine actively working to induce a docile state, the worm just stays aggressive, even without the octopamine gas pedal.
That is just a stunning piece of biological engineering. So the line between peace and violence is just this delicate arm wrestle between these two chemicals.
Exactly. But having the gas and the brake is only half the story, right?
Yeah.
A chemical is pretty useless if it doesn't have a receptor to bind to.
Right. Here's where it gets really interesting, because the researchers had to track down the receptors, like the keyholes for these chemical keys.
Yeah, they found two octopamine receptors, SIR3 and SIR6, that are required to make the worm aggressive.
The gas locks.
Right. And one tyramine receptor, LGC55, that stops it.
The brake lock. But it's not just about having the locks, it's about where they put them. In the peaceful C. elegans, these receptors are scattered basically all over the place.
Yeah, widely distributed across a large subset of neurons.
So it's like C. elegans uses these chemicals as a building-wide intercom system. The brain drops some octopamine and broadcasts a general fasting signal to the whole body.
That's a great way to think about it.
But in the predatory P. pacificus, they took those exact same keyholes and wired them directly into the security cameras at the front door.
Yes. In P. pacificus, these receptors localized to highly specific head sensory neurons, most notably the IL-2 neurons.
So what are the IL-2 neurons for the listener who might not be looking at a microscopic worm brain map?
Huh. Well, there are sensory cells that project right out to the very tip of the worm's nose.
Oh, wow.
Yeah, they are the absolute first point of physical contact with the outside world. When the worm bumps into something, the IL-2 neurons feel it.
So they essentially wired their sense of aggression entirely to their fingertips, or, well, their nose tips.
Exactly. The chemical is routed right to the part of the face that physically bumps into the victim.
And the researchers proved this, right? They didn't just guess based on location.
Oh, they definitely proved it. They engineered a way to selectively silence just the IL-2 neurons using a histamine-gated chloride channel.
Driven by the KLP6 promoter, right. I remember seeing that in the paper.
Yep. So they basically created an off switch just for the nose. And when they silenced those specific neurons, the worms essentially went blind to the prey.
Just completely ignored them.
Predatory aggression plummeted. They stopped biting entirely.
Okay, so wait. If the chemicals didn't change, and it's just a matter of where the antennas, the receptors are placed, does that mean this kind of aggressive rewiring could happen really quickly in evolutionary terms?
That is the big question. To figure that out, the researchers had to look back at the worm's family tree. They needed to see exactly when this violent rewiring actually took place.
So they went looking for the ancestor.
Right. They introduced a species called Allodiplagaster sudhausi.
That's a mouthful.
It is, yeah. It's a basal member of the same family as P. pacificus, meaning it's much older.
And just to be clear, this older worm is not just a peaceful grazer. It's like a giant cannibalistic monster, right?
It really is. It underwent a whole genome duplication event somewhere in its history.
So it copied its entire DNA playbook, which lets evolution experiment, basically.
Exactly. And the result is this massive worm that also has teeth and absolutely loves to hunt.
So what happened when they tested this ancient giant?
They knocked out the octopamine gene. They call it ASU-TBH1 in this species. And they saw the exact same drop in aggression.
Wow. So they literally counted fewer C. elegans corpses on the assay plate.
Yep. The gas pedal was broken in the ancient ancestor, too.
Yeah.
This raises an important question, right? It shows this isn't just a recent fluke.
It's an ancient evolutionary divergence.
Exactly. The entire family of Diplogastриdae learned to use octopamine and tyramine to gate their aggression millions of years ago.
Man, that is just incredible. To summarize this whole journey for you listening, we started by watching a machine learning AI sort through millions of tiny muscle twitches just to figure out what a hunt looks like.
Right?
We discovered that these worms aren't just hungry, they bite out of pure territorial aggression to secure their food source.
And they achieved this by rewiring their fasting and escape chemicals, octopamine and tyramine.
Yeah, taking them and plugging those receptors directly into the IL-2 sensory neurons on their face. It's just wild.
It really is. And it leaves you with a really profound thought to just kind of mull over.
Oh, I like where this is going.
Well, think about it. Evolution didn't invent a new aggression chemical for these worms. It just took existing peaceful chemicals and plugged them into a new socket.
The node neurons.
Right. So it makes you wonder if a simple shift in receptor placement can turn a completely docile grazer into a territorial killer. What dormant, complex behaviors are currently locked away inside our own human neural circuits?
Oh, wow.
I mean, we share a tremendous amount of basic neurochemistry with the animal kingdom. What's just waiting for the right evolutionary nudge to be plugged into a new socket?
That is deeply unsettling, but absolutely fascinating. Just a simple shift in where a receptor sits, and everything changes.
Yeah.
So what does this all mean for you? Well, it means the gap between peace and predation is biologically razor thin. It's all about the wiring. Thank you all so much for joining us on this deep dive into the tiny, terrifying world of killer worms.
It's been a blast.
And as a final reminder, please follow, like, comment, and share this deep dive with your fellow learners to keep the worm community thriving. See you next time.